Hello everyone, my name is Anastasiia Bondarenko, and before I start my presentation I want to apologize for not having a camera on. Unfortunately, I don't have one on my laptop, and the one on my phone didn't work on different online platforms. So yeah, there were some technical difficulties and I'm really sorry for that, but I do hope this will not affect the quality of the presentation. So let's get it started. Today I would like to present a very interesting paper called Predictable Maternal Separation Confers Adult Stress Resilience via the medial prefrontal cortex oxytocin signaling pathway in rats. Let's dive a little bit into the background first. Early life stress is usually associated with an increased risk for psychiatric disorders in future, and while some studies suggest that early adversity can enhance stress resilience in adulthood, the biological basis of this resilience is still unknown. Maternal separation, a common early life stress paradigm, has been shown to increase the risk for depression in adult mice. However, it can also trigger some adaptive processes that improve an individual's ability to cope with stress later in life. In particular, a predictable maternal separation is well known in terms of leading to stress resilience in rats. Regarding the oxytocin, it's known that oxytocin in the medial prefrontal cortex plays a key role in regulating stress resilience in response to early life stress. In this study, predictable maternal separation during the postnatal period was found to promote resilience to adult restraint stress, while unpredictable separation increased stress susceptibility. Differential gene expression analysis revealed oxytocin as a key gene associated with stress resilience, with predictable separation increasing oxytocin expression in the medial prefrontal cortex. So this study involved a timeline of MS from postnatal day 4 to 20 in rats, followed by group housing and subsequent behavioral tests, the results of which we can see on this graph. So the graph B depicts behavioral effects of MS and RS in sucrose preference test. Among control rats, UMS rats showed a decreased preference for sucrose compared to PMS and standard red rats. Among RS rats, sucrose preference in UMS rats was significantly lower than in PMS rats. Graph C demonstrates the heat map of average rat exploration during open field test. And as we can see, among RS rats, UMS rats and standard rat showed less activity compared to the PMS rats. According to graph D, UMS rats spend less time in central zone compared to standard rats, and PMS rats spend more time in central zone compared to standard rat and UMS rats after the restraint stress. Moreover, on the graph E, we can see the results of the elevated plasmase test, which showed that after RS, PMS rats spend more time in open arms compared to standard rat and UMS rats. On the graph H, we can see that PMS and UMS rats showed poor novel objective recognition compared to the control rats. But after the restraint stress, as we can see, PMS had an elevated discrimination ratio than other groups. So the tendency of rats to spend more time exploring a novel object increased. But at the same time, on the graph F, we can see something opposite, because the graph F depicts that no difference was observed in novel location recognition test. Interestingly, UMS rats were tending to choose relocate objects a little more frequent than PMS ones. So I think maybe the test itself was conducted in the wrong way, or there could be another reason for such results. It seems to me that stress doesn't necessarily lead to a lack of the desire to learn something new. I mean, we don't fully understand the mechanisms underlying the stress response, but we can assume that the presence of stress can also increase the interest in something new, because anything new is a potential opportunity to cope with the stress in future. But still, here it's not clear why PMS rats spend more time with the novel objects but didn't tend to choose the relocate ones. And as we can see on the graph G here, Among RS rats, social interaction ratio was reduced among UMS compared to PMS rats. And the last graph reflects the level of corticosterone. The concentration of corticosterone in UMS rats was significantly higher than in controls, restraint stress, and PMS groups on the first day of the restraint stress, and on the 7th day and after restraint stress, corticosterone in UMS was still kept in high level compared to other groups. So these findings highlight the complex relationship between early life stress and later behavioral responses in rats. On the graph B here, we can see the synaptic structure and postsynaptic densities of neurons. As we can see, UMS rats neurons have lower synaptic density, which is associated with depression severity. This is also shown on the graph C, which depicts that the length of postsynaptic densities are shorter in UMS rats than in control and PMS rats. Moreover, the depth of postsynaptic densities were shallower in UMS rats than in control and PMS rats, as we can also see on the graph D. In addition, graphs E, F, and G depict that spine density in UMS rats is less than in standard red and PMS rats significantly, and also UMS rats display less thin spines and mushroom spines than standard red and PMS rats. So such spine morphology means that UMS rats are experiencing anxiety-like behavior. These results suggested that PMS promotes stress resilience to restraint stress. There was also an effect of MS on mPFC amplitude. mPFC means postsynaptic response to the release of an individual vesicle. So a change in this amplitude is generally interpreted as a change in receptor number. So UMS rats significantly decreased amplitude compared to control and PMS, and at the same time MS had no effect on mEPSC frequency. They found that the peak amplitude of AMPA receptor-mediated postsynaptic currents was significantly lower in UMS rats than in standard red and PMS rats, which we can see on these figures I, J, and K. And to examine whether a dysfunction in presynaptic release somehow contributes to the observed synaptic defects, they also performed a paired pulse ratio analysis to detect any changes in the probability of presynaptic release, but no difference was detected, as we can see on the last graph. As we can see on figure A, UMS altered the expression of more genes than PMS did, and also there is a heat map of differentially expressed genes on the graph B, and differentially expressed genes analysis identified oxytocin, a peptide hormone, that has been shown to contribute to many aspects of social behavior, as the strongest key driver gene of these altered genes, which is reflected on the graph C. We can also see the results of RT-PCR, which show that the oxytocin expression in mPFC in PMS is higher than in standard rare and UMS rats, both before and after restraint stress. And also there are the results of the enzyme-linked immunosorbent assay, which demonstrate that oxytocin in mPFC in PMS and standard red rats is higher than in UMS rats before restraint stress. And after restraint stress, oxytocin level in PMS is significantly higher than in standard red and UMS rats. In addition, we can see on the graph G that the level of oxytocin in UMS rats serum is lower than in standard red and PMS groups 1. And so what the researchers did next, they injected the rats with oxytocin receptor antagonist and with oxytocin. Since PMS rats exhibited higher oxytocin levels in the mPFC, they tried to test whether the inhibition of oxytocin receptors in the mPFC could induce anxiety-like behavior in PMS rats. and we can see this whole procedure on the graphs H and I. So PMS rats injected with oxytocin antagonists spent significantly less time in the central zone and open arms, which demonstrated that the suppression of oxytocin receptors ability in the mPFC can induce anxiety-like behavior in PMS rats after restraint stress. And however, whether restraint stress was elicited or not, there was no difference in sucrose preference between OxyTR and saline-treated rats, showing that suppression of oxytocin receptor in the mPFC has no effect on depression-like behavior. And they also found that the application of oxytocin had no effects on duration in the central zone or open arm ratio in adult rats without restraint stress, which we can see on the graphs N and O. And it means that oxytocin microinjection did not improve the anxiety level induced by unpredictable maternal separation. However, both the time spent in the central zone and open arm ratio in rats injected with oxytocin were upregulated compared to rats injected with saline, showing that oxytocin in mPFC can improve stress resilience to adult restraint stress. And the sucrose preference level remained at about 60% in UMS rats even after the application of oxytocin, which indicates that elevated oxytocin in the mPFC was not sufficient to improve anhedonic behavior. Let's move further. To test the functions of oxytocin signaling in stress resilience more, let's say, accurately, they investigated whether suppressed OxyTR gene in the mPFC was necessary to promote the behavior associated with resilience. So yeah, the whole procedure is reflected on the graph A. The injection of AAV OxyTR shRNA in the mPFC knocked down OxyTR gene, reducing the expression of OxyTR by 60%. We can see this on graph B and also on graph C. And the concentration of oxytocin in the mPFC in PMS rats injected with the virus was significantly lower, as we can see on the graph D. Virus-injected rats displayed anxiety-like behavior in the open field because we can see decreased duration in the central zones on the graph E. And graph E and F, and this indicates that OxyTR gene knockdown in the mPFC of MS rats promoted a more vulnerable phenotype than control AAV scramble rats. In the standard rare rats, both AAV OxyTR shRNA and AAV scramble groups indicated anxiety-like behavior after restraint stress. However, the anxiety level in AAV OxyTR shRNA rats was more severe than that in AAV scramble rats. However, OxyTR gene knockdown in neither PMS nor standard red rats had no effects on sucrose preference test, which we can see on the graph G. And maybe it was because 3 days of restraint stress cannot induce depression-like behavior. To further explain the effects of OxyTR gene knockdown in the PFC, they also conducted behavioral performances 2 weeks later, and the anxiety level was still higher in AAV OxyTR shRNA rats than in AAV scramble rats, which we can also see on these figures E and F. And depression level was still the same between the two groups, which we can see on the figure G. In addition, OxyTR gene knockdown resulted in a reduction in mPFC frequency and peak amplitude compared to those of AAV scramble rats, suggesting that OxyTR knockdown altered synaptic transmission function. which we can see on the graphs H, I, and J. So these results indicate that reduced OxyTR gene expression in the mPFC shifted PMS rats toward a vulnerable phenotype. To determine whether the overexpression of OxyTR in the mPFC promoted stress resilience in UMS rats, they administered either control virus or OxyTR virus, which non-specifically overexpressed GFP or overexpressed OxyTR and GFP respectively, which we can see on graph K, in the whole procedure. And AAV OxyTR increased the expression of OxyTR by 50%, which we can see on the first figures, L and M. And interestingly, the OxyTR overexpression in the mPFC decreased anxiety-like behavior in the open field test, because we can see the increased duration in central zones on the graph O. This anxiety-like behavior was decreased before restraint stress, which indicates that OxyTR overexpression can rescue UMS-induced anxiety-like behavior. Restraint stress decreased the duration in the central zone and the open arm ratio in both AAV OxyTR UMS rats and AAV GFP UMS rats. And importantly, in UMS rats, OxyTR overexpression rats spend more time in central zones in the open field test than AAV GFP rats, which we can also see on the graphs O and P. And it shows that OxyTR overexpression promoted UMS stress resilience compared to the control group. The behavioral phenotype remained constant 2 weeks after restraint stress. However, overexpression of OxyTR in the mPFC had no effects on sucrose preference in UMS and sedentary rats, which we can see on the graph Q. And basal synaptic transmission measurements indicated that in the AAV OxyTR mPFC pyramidal cells, there was a partial rescue of mEPSC peak amplitude, but no effect on mEPSC frequency, which we can see on these graphs R, S, and T. So together, these experiments provided strong evidence that the scarcity of oxytocin in the mPFC of UMS rats accounts for its susceptibility to adult restraint stress. In rodents, neurons in the PVN are the main source of oxytocin projections to the brain. So to further confirm that the mPFC oxytocin signaling pathway is implicated in rat stress anti-anxiety behavior, they selectively prevented PVN mPFC oxytocin projections. The whole procedure is reflected on the graph A. And the idea was to achieve GM-cherry expression exclusively in PVN oxytocin neurons projecting to the mPFC. And they verified the regional specificity of virally mediated labeling of oxytocin neurons, which we can see on the graph B. Selective inhibition of oxytocin neurons projecting from the PVN to the mPFC was sufficient to reverse the PMS stress resilience phenotype, because we can see decreased duration in the central zone in the open field test on figures C and D, and aggravate the anxiety level in standard rats, because we can see decreased duration in the central zone in the open field test on figures G and H. The same tendency was observed in the elevated plasmase test, but the inhibition of PVN in mPFC oxytocin projection had no effects on locomotion activity, as we can see on the last graphs, E and I. So together, these findings show that PMS displayed resilience to stress by having increased oxytocin signaling, whereas UMS showed vulnerability to stress due to reduced oxytocin signaling. Thank you very much for your attention. Let's discuss this paper together. Please let me know in the comment section below whether you like this paper or not, and I will be glad to respond to all your questions and ideas next Thursday during our live online discussion. So yeah, have a nice day and goodbye.